Okay, everyone, welcome back. This is going to be our last video. It's going to be a lot more information as well. So please just bear with me. Let me go ahead and share our slide. And I just want to mention I'm going to put the links up to every video as well because now you were able to actually see my video, but you weren't able to hear it. So I'm just going to put those links up so you guys can watch them on your own. So now we're going to move on to basic microscopy. Let me make sure this is my first slide. Yes. So microscopes are utilized a lot in a laboratory, and they essentially aid the human eye by providing both magnification and resolution. So this is not using a microscope we're going to discuss today. This is probably using a confocal microscope with fluorescent indicators, if I had to guess. But essentially, as you can see, one, this is very magnified. We would never be able to see this structure. This is probably a DAPI stain in the center. So the nucleus is stained here, or nuclear material is stained here. We would never be able to see this with our eyes. Our naked eye, we would need a microscope to see this. And then you could also see how much resolution we get. This is very out of focus, and this is a nice focused image. So resolution is necessary to see details in your specimen. Oh, and here's the note saying that these images were taken from a different type of microscope, not the compound light microscope, which you guys will be using virtually. So the role of the microscope is to provide both magnification of an object as well as resolution. Resolution provides contrast and the ability to distinguish detail in materials being observed. There are several types of microscopes used in biological observation, and they differ on the basis of energy source and lead system. Usually, multiple microscopes are used together to answer scientific questions. And I touched on this in our previous lecture last week about the dissecting microscope, also referred to as the stereo light microscope. That one is usually your step one in a series of microscopes. So I'm going to first review the electron microscope. This is a very sophisticated microscope, and there aren't any activities in your lab manual that you have to complete. And most laboratories do not have this microscope. I was actually fortunate enough to get to use one during my PhD candidacy, and this image at the very top is actually from my paper. But they're very expensive and they're pretty rare to use. So these microscopes use a beam of electrons as the energy source. Their resolution provides 0.0022 to 0.005 micrometers, and the magnification is up to 500,000 times. So this is super magnified. In transmission electron microscopy, electrons are focused by electromagnetic coils. Electrons travel through the specimen at different rates, giving a high-resolution, one-dimensional picture of the cell. So these are all going to be one-dimensional images. Your confocal microscope can give an X, Y, and Z plane to your images. This one's going to just be a one-dimensional image. In scanning electron microscopy, electrons bounce off of the surface of a specimen, giving a three-dimensional image of the cell surface. So this is a transmission electron microscopy. This is scanning electron microscopy. So it really depends what tool you're using. In the laboratory I worked in, we only had access to the transmission electron microscopy. So that's why you see these one-dimensional photos versus a three-dimensional image. So here what you are seeing is the close-up of this image here, essentially. So in your brain, or your central, let's say in your brain, you have these cells called neurons. These neurons are very powerful cells, and they have dendrites that receive these chemical messages. And the chemical message will travel to the cell body or soma, and then it'll become an electrical signal where it travels down the axon. of the axon is properly myelinated, so it has the proper insulation it needs, it will carry this message extremely fast. In some neurodegenerative diseases, this axon, this little fatty tissue around the axon that makes it travel, the message travel super fast, will die off. So that's common in multiple sclerosis. So the message will travel slower. Or in axonal neurodegenerative diseases, this axon just starts to die off and just shrinks till there is no axon. So on the top, imagine this is a straw and you just cut the straw and then you're looking at it from the circular view. That is what you're seeing here. Here you're seeing all axons from different groups. So my untreated, this is a healthy mouse. You can see that there's a lot of axons. The axons are illustrated as this black material around the axon is all of this, but this black material is that fatty tissue I was talking about. It's called myelin. So as you can see in our untreated group, there's one, a lot of axons, and two, they all seem to have this fatty tissue around it. Here, these animals were experiencing neurodegeneration, and this was actually our experimental group here. And as you can see in this image, there's less axons, and when there are axons, they tend to not have that black rim around them to the same extent. So that's basically what I was showing there, but it shows you such good resolution, and you can even see the organelles in these axons. So you can see the mitochondria and the ER, all kinds of organelles inside these axons. That's how high the magnification and resolution are. So this is essentially how I conducted these experiments, just showing you the different microscopes you use. So you get your mouse brain and you use this microscope we talked about last week, which is called the dissecting microscope, to dissect the area of interest. So I used an area somewhere here. It's not important. But then I slice that relatively thin, and I look at it under a compound light microscope, which is this microscope here. I stain it so that I can see the contrast, and then I just make sure, okay, I'm in the right brain region. Once I confirm the brain region, I even slice it thinner, so much thinner than what you're seeing here. We actually slice it with a diamond in order to get it so thin. It's called a microtome that we use. And then you go to use this microscope. This is your transmission electron microscope. And that's how you can obtain these images now. And that is really just cutting this, a cross-section of this, and looking at the circular portion of the axons. So another way you can use the scanning electron microscope. So now this is, remember that 3D image that we were talking about. You can use a scanning electron microscope and you can put different attachments to it. So this is a SEM EDX microscope. So the EDX stands for Energy Disperse X-ray Spectrometry. And what I'm showing here at the top are particles, individual particles of gunshot residue. So this is how you would use this microscope in a crime lab. So this microscope would be great at getting the showing the morphology of the particle. So it's typically spherical, but it can also be irregular. And the spherical particles are typically 0.5 micrometers to 5.0 micrometers in diameter, so that is extremely small. And those that are irregular shaped are 0.5 micrometers to 100 plus micrometers, which is still extremely small. So here you can actually see the morphology of these individual gunshot residue particles. You can also see that at one point they were molten. And then this EDX portion of the microscope allows you to do an elemental analysis. So now you can see what these GSR particles are composed of. So you basically this microscope uses three detectors to composite this image you see here. It's going to use your X-ray detector, your backscatter detector, and your secondary detector to compose this image here. And it'll show you what elements are found in this particle. So say this particle gets brought into your lab and you don't even know if it's gunshot residue or not. First, you're going to look at the morphology using the scanning electron microscope. And then using the EDX addition, you're going to be able to see what this particle is composed of. So then using this EDX portion, you're going to see that this questionable particle is composed of, I can barely see this, sorry. It's going to be composed of lead, antimony, and barium. And those are the three elements that you find in GSR. So GSR is known to be composed of those. So then you can really say like, yeah, this is a GSR particle found on this individual's clothing or individual's hands or whatever it is. So that's another way of how you can use this microscope in a laboratory setting. And it's quite powerful and it gives you a lot of information, especially for trace evidence. When you really aren't sure what you're looking at, you can get a lot of information using this microscope. Like I said, both the scanning electron microscope and transmission electron microscope are extremely expensive and really rare to use. I was just really fortunate to get to use one in grad school, but they're not very common. What you typically have in a lab would be a compound light microscope. You are going to be expected to know the anatomy of this microscope. So up here you have your oculars or your eyepieces. This is what you're gonna look into to be able to view your your specimen, I'm sorry, your specimen. So you're gonna have to adjust these oculars to the appropriate width of your face. And these oculars will give 10 times magnification. This is your arm of your microscope. So if we were in lab, you would have to carry your microscope by the arm and the base. This is the on and off switch. This would control your brightness. Your light is going to shoot out from here. This is called your diaphragm. There's a lever on your diaphragm that you can control how much light you allow to pass by. So you have an aperture setting here that you can control. So if you're in photography, you understand that aperture is how open you control how open or closed you can make this. So the more open, the more light you're going to allow to pass through. If you close it up, less light can pass through. Here you have your X and Y axis that will move your stage. So this is what we call your stage. This is where you'll put your specimen slide and you'll clamp it in. I'll show you a video how to do that properly. And your stage can basically move from the X axis, so it can move left and right, and it could move to the Y-axis, so in this direction. It's always going to be on a flat plane. The way your stage will move up and down is through the coarse adjustment and fine adjustment knobs. The coarse adjustment knob is going to move that stage quite rapidly, so you are only supposed to use the coarse adjustment knob when you are using your 4x objective lens, which is shown over here, the little lens. When you are using higher objective lenses, you can only use your fine adjustment knob. The reason is because the coarse adjustment knobs move so quickly that you can damage your lens and your specimen, actually, both of them. You'll damage your microscope and your specimen if you crash your slide against the lens. So here are your objective lenses, and this is called your nose piece, so you can move this around to get it on the correct objective lens you need. You have 3 objective lenses shown here. Sometimes you will also have more. But here we have our scanning power objective lens, which is our 4x. It will provide less magnification. Then we have our low power objective lens, which is 10x. It's shown here in yellow. And then we have our high power objective lens. This is going to provide the most magnification, and this is shown here in the blue. Remember, when you are using your 4x objective lens, you can use both the coarse adjustment and fine adjustment knob. When you are using your higher objective lenses, so either your 10 or 4x, I only use the fine adjustment knob. I never have a need to use the coarse adjustment knob on those lenses. But just go through, make sure you're familiar with all these, the anatomy of your microscope. You know how to identify all these pieces. If you have questions, let me know. So if we were in lab, you would have to know how to properly carry your microscope. So basically, this is a no-no. You want to carry it from the arm as well as the base. So we're first going to cover magnification. So your oculars will give you some magnification. This is going to be 10x magnification you're going to get from your oculars. Your objective lens will also give you magnification. So if you are using your 4x scanning lens, it would be giving you a 4x magnification through your objective lens. So the way to calculate total magnification is to take both of these into consideration. You need to multiply your ocular magnification by your objective magnification. So your total magnification will be oculars times objective lens. So if you are using the scanning lens, which is your 4x, you would do 10x from the oculars times 4x from the objective lens to give you a total of 40x total magnification. If you were using your 10x low power lens here, you would use 10x from the oculars times 10x from the objective lens to give you a 100x total magnification. So remember, you're always going to take both into consideration for total magnification. So this just kind of goes over magnification again. And we're now going to talk about fields of view. So pretend you're using a microscope and you're using 10x ocular magnification, so on your eyepiece, and you're using a 4x scanning lens, objective lens. So you're using the red one. You're using that on your microscope. You have your specimen on your stage and you grab a ruler, probably like a plastic paper, very thin ruler, and you decide to measure the diameter of your specimen. And you see that the diameter of your specimen is 2.5 millimeters in diameter. When you're at 4x objective lens, it's pretty easy to measure your specimen. As you increase your objective lens, you're going to notice in 2 slides from now that it's more difficult to measure your specimen because your working distance is reduced. So when you're at 4x objective lens, it's pretty easy to get in there with a ruler and see how big your specimen is. So pretend you're using your 4x scanning lens. You measure your specimen and it's 2.5 millimeters. What is the diameter with the same ocular and a 10x objective lens? So our field of view right now is 2.5 millimeters. We are using a 10x ocular magnification times our 4x objective lens magnification for a total of 40x magnification. But we want to know what if we went up to our 10x objective lens. Now we're going to be at 100x magnification. So you're going to use this formula to determine this. So you're going to it's going to be your high power field of view over your low power field of view equals your low power magnification over your high power magnification. So just go ahead and fill this in. So we don't know our high power field of view. That's what we want to determine. So that's going to be X. Our low power field of view is 2.5 millimeters. So we're going to plug that in here. This equals our low power magnification. Our low power magnification is 40x. So at 40x, our specimen is 2.5 millimeters in diameter. And what we want to figure out is our high power magnification and what the diameter is at our high power magnification, which is 100x in this example. So you're going to cross multiply. So 100 times x equals 40 times 2.5, which is 100. So 100x equals 100. We want to isolate our x, so we're going to divide by 100 on each side. So x equals 1 millimeter. So our high power field of view would be 1 millimeter. So what does this mean? When we are using a 40x total magnification, our field of view is 2.5 millimeters. When we change our total magnification to 100x, our field of view is only going to be 100 millimeters. So when we go up to that higher magnification and we look through our microscope, we're only going to be able to see probably this part of our specimen. When we are looking through the microscope at 40x, we can see all of this of our specimen, you know. At 100x, we can probably only see this portion of our specimen. So what this means is as magnification increases, your field of view decreases. This is the same example. So now imagine you increase your 40x objective lens for a total magnification of 400. What is your field of view now? So instead of 100x here, we're going to plug in 400x. We're going to solve, and that answer would be 0.25 millimeters. So when we are using the 40x objective lens, we're probably only going to see a very small portion of our original specimen, like this much of it. So basically what this is showing is that as magnification increases, your field of view will decrease. So once again, when we were using our 4x objective lens, we were able to see 2.5 millimeters of our specimen. When we moved up to our 10x objective lens, we were only able to see 1 millimeters of our specimen. When we moved up further to a 40x objective lens, now we can only see 0.25 millimeters of our specimen. So our field of view is going to get very small, but we're going to be able to see a lot more detail, and that's why it's important to move up in magnification, because you will get more detail out of it.